and go over is kind of getting you to think about why we should transition our lab courses to integrate research to become these course-based undergraduate research experiences, and thinking about how to actually assess those courses. So first, over the past 10 years or so, there have been a number of national recommendations that have promoted how important it is to integrate undergraduates in research. And so some of these national reports have basically said that every single science major should go through some kind of research experience as part of becoming a science graduate. Participating in undergraduate research has been shown to increase students' odds of lots of things. So persisting in biology, obtaining a bachelor's degree, expressing interest in pursuing a PhD, pursuing a STEM graduate degree, and actually entering a STEM graduate or professional program. If we look at science graduate programs, particularly more elite science grad programs, undergraduate research is essentially a prerequisite now. If you don't have undergraduate research, then you're not going to be able to get into those science grad programs. And if you think about it, it becomes kind of an equity issue, because then if you don't get into those more elite graduate programs that often have more funding and more opportunities, then your potential for kind of being able to persist in science might actually be less, right? And so these are things that these schools don't often come out explicitly and say that someone has to have it, but if you look at kind of what their expectations are for admissions, they talk about needing to see undergraduate research. Typically, when we think about undergraduate research, we think about students working in faculty members' research labs. So sometimes these are called individual research apprenticeships, sometimes they're called individual research experiences. So these can take the form of summer REU programs, right? And so that is kind of most common. But there's a number of problems with this model. So obviously there's a limited number of positions available in faculty members' research labs, and at most institutions there are way more students enrolled than positions available, right? So for example, at my institution, we've got 1,000, we have about 700 actually incoming biology majors, right? And we've got about 100 faculty, right? And so even that's just one class, right? And there's no possibility that every student can get involved in a lab. There's also, because there's not enough positions and way more students, there's often a selection process for who gets to actually participate, right? And there's, students have to actually know what people are starting to call the unwritten rules of undergraduate research. So they have to know that undergraduate research is this thing that's good. They have to know that to get involved, you often just email a faculty member. They have to know what to write in that email to the faculty member. They have to know what to dress when they go in and see that faculty member. They have to know that they have to pretend to be super excited about that topic, right? And there's going to be some students that know these things, and there's going to be other students that don't know these things. We actually have an ongoing project in the lab where we're trying to actually identify these unwritten rules and then be able to make these rules more explicit to all students, right? But you can imagine that students from more privileged, students that have parents who are academics, they're going to know these rules. Other students, not so much. Students also have to have that confidence to apply. They have to know that research is a thing. They have to know those unwritten rules, and then they have to actually take that step and go out kind of the normal, kind of confines of signing up for classes to take that extra initiative and email a faculty member, and they have to add that time on, right? And so that is going to factor into the selection process. Further, the faculty pick, right? And faculty are incentivized to pick the best student, right? So there's very little incentive in place for the faculty member to pick that student that could have the most growth potential, that student that could be struggling. So faculty use GPA, they use their grade in intro bio, they use how similar that student aligns to white culture. They use all kinds of different factors to pick that best student, and there's not very many incentives in place to take the student who could take extra work, right, but could actually really benefit from the experience. Then there's also been ongoing studies that have shown that faculty, like everyone, have implicit biases that may advantage certain populations of students. So studies have shown that faculty are more likely to respond to emails from white-sounding names compared to black-sounding names. And then there's a study that shows that when students apply for a research assistant position in a lab, faculty are more likely to hire men, pay them more, and mentor them more, right? And so these implicit biases could actually factor into who they're actually choosing to participate. So there's there's kind of two problems with this model of students engaging in undergraduate research and faculty members' labs. Only a subset can participate, and these students who participate tend to be from more privileged backgrounds. So I want folks to turn to someone kind of at the table or a nearby table, and what are solutions to this, right? How do we solve this problem? If research is so important, right? And this is the model.
Awesome.
Hi.
Cool. All right.
My thought is almost kind of like a lottery type, like matching or grade system, where maybe people talk about their interests, but instead of having kind of some of those other selection factors like their GPA, one kind of their goals, so they just kind of get one interest, and then maybe everyone like has a desire. There's a way to do that, but that would obviously take a lot of like self-assessment. Right.
Starting point, maybe. I can see that. The other thing is, which is not where she's necessarily going, but periodically, over the time I've been here, there's been plans that have been set for engagement with students
One or two things
don't
going
come
on
from
right now that are.
early career, under-resourced students, so that there's an incentive for a faculty member to take that research course. They come with money, right? And so, she's saying there's no incentive for a faculty member that's got a limited amount of space and a limited amount of money.
And then I never exposed
take a
myself to
risk,
things that maybe there was
or
someone
what would be perceived as a risk on a student that doesn't seem like the best student.
Right.
So, and one way to put it is maybe to programs specifically incentivize faculty.
Making a little bit of it like kind of blind.
That doesn't solve the
Some of the
first
things
thing,
that may cause
because it's not, that doesn't solve the capacity.
So about 30 more seconds to finish up your discussions.
Yeah, I was gonna say, what if we started a class and they all take it as undergrads? We extend them. So basically,
Sometimes.
yes, we got your
Okay, so let's go ahead and come back together. So what are ideas? So what are some possible solutions? So stuff that you do, stuff that you could do, that you don't do.
So this doesn't solve the first part, but the second part would be to incentivize faculty to take other kinds of students that are maybe non-traditional or not the best students. So having pools of money to change incentives.
Yep, so maybe pools of money specifically for underrepresented students or non-traditional students or transfer students or kind of any of those groups that are potentially less privileged, right? And NSF does this, right? They have pools of money specifically for populations of students that could help diversify who gets to grow up to be a scientist.
And so I have students doing independent research projects in an upper division course that I run, so I do both the lab and the I reckon the PA for that. So, but the challenge is that I have to be in control of both the lab and the lecture in order to like implement this. And the second is that because it's upper division, I've already lost subset of more marginalized groups because they've already dropped out. And so it's okay, it's better than the 2 or 3 that work in my lab, but it still is taking a subset and primarily more privileged.
Yep, so you can integrate it into courses, kind of how I was starting off the talk. You can give the opportunities to more students, right? But depending on what courses you're doing, upper level, you've already lost students at that first year level. Is it 20 students, is it 50 students, is it 100 students? Like, what's that scale of that, right? Other ideas? Yeah.
So I work at a faculty development unit, so I don't teach, but I think the first step is to make faculty aware that bias exists. And once they're conscious of that, they will make their practices change accordingly. Because I'm sure nobody has come here with a desire to exclude people. But sometimes, they just don't know that such biases exist. I think that awareness is.
Yeah, just trying to make your bias more apparent, right? Especially for individuals who maybe didn't grow up in as diverse communities, they may not know different cultural differences, right? And I know that I have experienced this as a mentor, realizing that I was assuming that students sometimes weren't enthusiastic if they didn't display the outward behaviors that I valued as being enthusiastic, but actually I realized over time that a couple students, they were enthusiastic, it's just their cultural backgrounds had them display their enthusiasm in different ways, right? And I had to kind of readjust my own biases, right? So we can take kind of the current system and we can say, okay, how can we make it so that we can more equitably distribute those opportunities? How can we get faculty to be more conscious of their own biases? We can also be conscious of our departments or our universities, what policies exist that are potentially biased, right? So a concrete example, we have a research kind of scholarship program for students at Arizona State University where there's a minimum GPA of 3.0 to enter. But yet the funds for this program come from student program fees. And so this has been a real point of contention for me because I'm like, everyone is participating in the program fees for this program. They submit an application, and an advisor has to kind of sign on and say, this student, I want to work with them, etc. Why do we have a GPA cutoff, right? So a student, if they have a 2.9 GPA, they're not going to be a good researcher, they can't be a good researcher. You know, we have very little evidence that supports that kind of a high GPA is going to be a good predictor, right, of a researcher. And so I think we can begin to think about kind of what policies are in place in our departments that are potentially inadvertently causing just more privileged students or more academically kind of adept students.
Can I ask a question about that?
Yeah.
So I have been trying to decide, and I'm just starting research group, like whether I should ask people who want to join to share with me their transcripts. And I was one of the low GPA students. When I was an undergrad, I had to work 30 hours a week and go to school full time, so I had a I feel like a legitimate reason. And I kind of have been juggling whether or not I should ask for the transcript because I feel like it opens the door to have a conversation about where they're currently at and how they're doing in classes. But I also don't want to make it awkward. I have a hard time sort of demanding that.
I mean, I think it's.
What do you think about that?
I think it's dependent on what you want to do, right, and how you use that, right? So personally, I don't ask for transcripts for students because I realized that I wasn't going to use that in my assessing of the student about their possibility of being a good undergraduate researcher. But if that is going to lead to a conversation that then you're going to, you know, assess some quality of the student through that discussion, you know, then I guess
I see myself as a stepping stone for a student to move on in their life, and whether or not I care about a GPA, somebody else is. And so I feel like if they have a low GPA, I can talk to them about it. We say,
Sure.
how do you help to get there? But I still feel like it's a personal thing. I don't necessarily want to share my transcripts with people.
Yeah.
Still.
Yeah, I think that most students are probably okay with sharing their transcript or their GPA with faculty because I think that that's often something that gets asked for. You know, but I think it's important to look for other characteristics that may be better predictors of a student's kind of aptitude for research and their interest in research, right? Because otherwise you're potentially, you know, blocking those lower performing students from participating, right? And if this is something that everyone should participate in, then well, we're only giving that kind of upper group of students that extra experience, right?
We use federal work study to employ students to do various things, and I've got programs that do community outreach and citizen science, and so, you know, Pell eligible students generally are the ones that get opportunities for federal work study, and so I've used that as a model to engage students in undergraduate research.
Yeah, so then picking up on kind of a separate mechanism that can try that. So a lot of what's been done over the past, you know, 10, 20, 30 years is creating initiatives to diversify, right? So NSF programs trying to kind of create those solutions. But as Susan hit on, that's only going to address that bottom problem, and that's not going to address that top problem, right? You're still only going to have a subset of students that can participate. Even if faculty are incredibly altruistic, there's still way too many students for every student to get an opportunity to do research, right? And so so a different solution as far as integrating research into undergraduate lab courses could actually attack both of these problems, right? Especially if you build research experiences into introductory courses right? And some people, and some institutions have done this where it's hundreds of students are participating in research as their almost first exposure to what a lab course looks like in college. And so that way every student that graduates with that degree actually gets that opportunity to do research. So people use all kinds of different terms to describe these courses. I tend to use the term CURE, or course-based undergraduate research experience. Some folks call these course-based research experiences, and they prefer that term because these could actually be graduate courses as well as undergraduate courses. But CRE isn't kind of as catchy as CURE, and so I think that's why CURE is caught on more. Sometimes these are called discovery-based courses to emphasize that novel research element, and some people have called them kind of research-based courses to emphasize the research. And so trying to find literature on these courses is kind of challenging because people are using lots of different terms to describe these, although the community as a whole is moving towards the term CURE, I would say. So in thinking about how CUREs can address those two problems, a colleague of mine who's a community college faculty member and I wrote an essay a number of years ago highlighting how CUREs can make scientific research more inclusive by addressing those issues. That if you develop high enrollment CUREs, you can increase the number of research opportunities available at your institution, increasing access, and importantly, requiring students to enroll in a CURE as part of their normal curriculum. It gets rid of them having to know those extra rules or those extra kind of decision points and that extra confidence. Faculty don't have to select students, and so students sign up for the course just like any other course. Faculty teach them just like any other course, and so it removes some of those barriers to participation. So course-based undergraduate research experiences can increase the number of students who participate and be a way for every undergraduate to have a research experience, right, depending on how you actually structure it. So a few years ago, so what makes a CURE an actual CURE? So a few years ago, a group of experts who think a lot about undergraduate research experiences and CUREs and lab courses came together and they identified what they thought were the critical elements of a course-based undergraduate research experience. And these included scientific practices that students were going to work on problems that scientists work on, right? And that they were going to engage in practices that scientists engage in. So communicating their results to each other, analyzing data, designing experiments, actually doing experimentation, et cetera, et cetera. They identified that collaboration is often an important part of research, and so they wanted students in these CUREs to actually be working together to build on each other's knowledge, and so to exist in kind of real collaboration. Iteration, the idea that you can't just do an experiment once, that you actually have to repeat it, that you have to have multiple replicates, that you actually have to build on prior work of someone else, and so getting students to realize that there's published literature that they need to be aware of and have read to then build on to something that's new. And so I'm thinking about iteration not just in terms of the student's own hands, but also in terms of how that research project is kind of iterative based on what's been done before. And then the last two is discovery and broader impact or relevance. So discovery highlighting that what the students are working on is truly novel, that people don't know the answer, and that there's relevance to people outside of the classroom, that someone cares about the actual results. So they proposed these 5 dimensions. They didn't know a priori about whether or not these had to actually be dimensions of CUREs, but this was a way for them to first start to kind of conceptualize what makes a CURE a CURE. I like to use these to think about how CUREs are different than other types of lab courses to help get people kind of on the same page. So if you think about kind of traditional cookbook lab courses, cookbook because students are kind of following a series of steps, much like a recipe in a cookbook, these courses tend to focus on scientific practices, often pretty low-level scientific practices. Can students follow a series of steps? Can students get the right answer? If they compare it to what they should have gotten, can they explain why they didn't get the right answer, etc.? There's sometimes a tiny bit of collaboration in these courses, but usually it's inauthentic. It's we don't have enough beakers to go around, so you guys are going to share your beaker, right? There's usually no iteration. Often these courses, every week is something brand new. Often these are aligned with the introductory or with the actual lecture course, right? And so usually they do something once and that's it. No discovery and no broader impact or relevance. Now, inquiry-based activities, and the term inquiry is kind of fraught with issues because people use inquiry in lots of different ways. And so the way I'm using it here is to describe the types of activities that students might participate in the lab course where it's novel. They don't know what the answer is, so they're doing something completely new. So kind of the classic inquiry experiment that we do in biology courses is you might have students go and swab doorknobs to see kind of which doorknob has the most bacteria, and they can make hypotheses about which doorknob, and a priori we don't know which doorknob has the most bacteria, right? Now, maybe there's broader impact or relevance there, but not really, right? You're not going to write a paper about, you know, which doorknob in this particular building has the most bacteria. But for the students, it could be a really important, compelling experience, right? Where they can really start to grapple with how to create a hypothesis and how to think through kind of a novel result and how to compare, you know, what they were predicting with what they actually got. These types of courses tend to involve a little bit higher level scientific practices. They're often much more about students designing their own experiments or analyzing data. But often these types of courses are set up so that they're modular, where there's not a whole lot of iteration in these courses. But what really makes Cures distinct kind of from other types of lab courses is this combination of discovery and broader impact or relevance. And really what distinguishes Cures from how I'm distinguishing kind of inquiry-based activities is that the discoveries that students are doing in a Cure, someone actually cares about beyond the scope of the course. That there's kind of novel findings that have the potential for an impact. In fact, I've argued, and other people have now corroborated this, that this is not actually two separate ideas, that this is actually one single idea. And so the kind of the magic sauce of a Cure really is that what the students are working on is real research, right? So it's analogous to the type of research happening in faculty members' labs. And so we need to make sure that that's actually built into the Cure. So how do we know if it has an impact beyond the course? Well, the lowest level is that the kind of most conservative way is, does it result in a publication, right? And there have been lots of examples of Cures where students have collected data, analyzed data, and it's resulted in a scientific research publication. Sometimes they're co-authors on these publications, sometimes the class is acknowledged, and there's lots of debate in the community about what should constitute authorship and whether a student in a Cure should actually be an author. But we all know that there's a lot of steps that have to happen to actually get to a publication. And so many Cures are set up with more intermediate goals. So it might be adding something to a database that other people can access. Sometimes that database might be a national database that anyone could access. Sometimes that database is actually just a database for that faculty member's research lab that then the graduate students and postdocs in that lab could access and build onto. Or depending on the question, maybe a publication is not the most appropriate final product. So maybe if you're measuring kind of water quality of a local stream, you actually want to write some kind of community report up that could change people's behavior, that people potentially could care about, but it wouldn't be like a scientific research publication. Okay, so all that said, and I'm kind of, you might say, so why am I belaboring kind of what a Cure is? There's a lot of people who really struggle with what makes a Cure a Cure. And in talking to faculty kind of across the country at multiple points, I think it's tricky because Cures have become this like exciting thing for faculty to do, and so sometimes faculty want to consider what they're doing a Cure even if it's not a Cure. And so my disclaimer is, I'm not at all saying that Cures are better than inquiry experiences. I actually think that they could have potentially very different goals, and I'll talk about that a little bit later in the workshop. But I do think it's really important from a design and an assessment perspective that we are talking about the same thing when we're talking about kind of inquiry versus Cures. And so that's why I'm kind of belaboring it here. So I thought that it could be great to kind of wrap our heads around this, to think about these 5 scenarios and debate with folks at your table about whether you actually think that that's a Cure. So if students were participating in these situations in a class, would that be a Cure? So go ahead and turn to your neighbor, just get loud here.
Okay, so anyway,
Yeah, I think
That's a challenge. It's like
and that was how it
fill it out ahead of time.
It's just being in the lab and getting a problem that says, what is the answer to this? No.
In other words, that's like they're designing something, they're thinking about something they can do, but they're not actually generating it.
So, that's the phrase, a lot of things, like, we have a general idea of how they work, but then there's like, what about this mechanism? So I teach computers at Sara Brownell,
In my
fire technology, and restoration, like evaluating restoration practices.
maybe a little bit more
We have a general idea.
data to push into the
What is over in cheatgrass as well, but there's a lot of different methodologies.
Well, but it does,
What are the
I mean, it does use scientific practices to do this analysis.
struggle with?
Well, but it could be something other here. Right. And I think
It's like, how do you fit them into like your research agenda to make them, so
that's like
where broader impacts relevance. Why do people care?
So I would
But
agree
I
with
do
you,
still value those
but
experiments like
the other cure that I
familiarizing them with
super Colorado
techniques. But I think in this is a connection to yeah. It's like if I package it up.
Colorado,
Right.
what is it that see of this thing? Because I could go down to the shop and get whatever, and,
they don't show any variation.
but you know, then you yeah, exactly. So then like finding that relevance, so, you know, like, but then I always think about like, well, then tying it back to like, what's the scientific relevance? You know, a lot of students have this question, who cares?
Right.
And
And you could imagine that, you know, it's in a data.
you're right. So but then it's like, well, then you need to build that bridge. Yeah, you need to build that bridge, so it's like,
Now they don't have to test this, because they know,
you know, some students have different questions. Because, you know, how does
you might not
attaching
know.
an egg sac from
So that could be
survival
survival.
So that's a great
would that ever happen in nature? Like, why would the egg sac not attach? Like, they're meeting that first scenario of like, nobody knows, but like, has really no relevance. So then it's like, well, how do you do new and novel things that are relevant?
And then you just do something related to test it, efficacy of different methods to now let's and then maybe that would like cures.
That's a really great point, yeah.
So I think that's like
So I agree with you,
their,
if it's not,
yeah, a novel question, like if there wasn't a big research that that kind of had the answer.
Like, because of that, they're coming up and, but I think that's not.
what do you really do now? Like, I don't really understand. You know, because they're like, we developed this thing, and I'm like, so it's like just an idea, right? And you're like, and you spent 16 or 10 weeks on that.
2007, and John Mark was good, and I was here for 3 months.
Maybe my question is like, what about math?
Back in Holy Economy,
Then beyond that.
you know, bubble was person. So yeah, it's just one of those things where you have these goals in mind, but then it really comes down to, you know, who's hiring and just research put in the door.
I'd like you to have data science. It's kind of like.
All right, so let's go ahead and come back together. So can I get a brave volunteer to identify one of these scenarios and whether or not you think it's a cure and why?
We went for no on this one because it feels a little bit more that they're leaving out kind of the part that's the like broader impact, relevance, or knowledge of prior research and scientific advancement.
Okay.
So that to make it more of a cure, you would have to expose them.
And which one? To which scenario? Sorry.
To the scenario one.
The
So
scenario
expose them to
one,
the answer that the scientific community already knows and then go from there.
Okay, so you don't think scenario one is a cure?
Yeah, we don't know.
Did everyone agree with that?
Yeah, we said no, we put an asterisk.
Okay.
But, with the question, does the scientific community actually always know the answer?
Okay.
And so I think, like, depending on the situation or the system, or maybe it's a slight modification, I think there's some value to contribute. Because you might find in your particular area that you don't get the same outcome, and then you can explore why.
So we had that same discussion, is it just validating what's already known, or are you actually like exploring something that nobody really knows for this particular system that might fit into that broader context?
Yeah, I mean, there's some things that we know as a scientific community, right? But there's a lot that we think we know, right? And then a new technology appears and we realize that we don't know it completely, right? Or a couple years ago, there was a lot of debate about psychology experiments that are unable to be replicated, right? And so just because kind of the academic or scientific community thinks that they know the answer, just because something's been published doesn't mean, right, that's necessarily for sure. And so I think actually a really interesting way to use cures could be to actually replicate, previous studies. And so in some ways it's a confirmation experiment, but in other ways it's seeing if an experiment can actually be replicated in a way that you wouldn't necessarily want to put a graduate student on it because it's not going to lead to a really exciting paper, but it could be a way to build in a cure for students, right? But that would have to probably be communicated to them, that they realize that they're not just, there is no right answer, right? This is what the scientific community thinks, right? And then the goal of that cure is to see if that can be replicated or not, right?
So it might depend on how you frame it.
Mm-hmm. Yeah, and it also might depend on how foundational that particular thing is, right? So if it's completely foundational, then it wouldn't be, it wouldn't be. What about another scenario? Yeah.
We talked a lot about scenario 4, and I think we came to the consensus that we think it would be an example of a cure, but the part that it's obviously leaving out is the data collection aspect of that scientific practice. But we talked about the fact that there are graduate students that do this and they're doing real research, so we thought that yes, this would be an example.
Yeah, absolutely. I would say that that's a cure. Now, I think it's a really interesting open question. What is the impact on students of actually collecting their own data, right? Do they have greater project ownership by collecting their own data? And that is a question that we don't know, right? And that's a question that I'm super interested in, other people are super interested in, because then we can start to make decisions pedagogically in our cures, right, about whether we can actually have a bigger impact on students if they're collecting their own data or not, right? But maybe it doesn't matter, right? And maybe it depends on the data set, right? Because you could have data sets that are just really cool that students couldn't have possibly collected, right? So I have a colleague who does awesome biology in space, right? And students aren't going to go up into space and grab that data, right? But then if they could actually use that in a cure, that might be super compelling for them, right? And so, yeah.
And I think that actually links with number 2 of the thought experiments. And I mean, I see this as computational opportunities of virtual modeling that brings things together. And I think that might be seen as you can collect the data, but we have real scientists. That's what their research programs are, is they take existing data and or simulate things with virtual organisms and say what should happen. And so making that connection though remains that same issue of how do you make sure that students sort of understand where that's coming from from a scientific perspective.
Yeah. Something that I think people are becoming really interested in the cure kind of community is to what extent we're being transparent about what the students are doing, how what they're doing is real research, how a specific component of what they're doing is actually research. Because I think often students assume that lab should be experimentation, right? And anything that they're not doing that actually is moving or manipulating something with their hands is not necessarily lab. And obviously that's most of what we do is actually not the manipulation, right? It's thinking about the project beforehand, it's analyzing the data afterwards, et cetera. And so just making that transparent to students might need to be an important component of cures that right now I don't think is happening to the degree that it probably needs to. So yeah, so I think that a lot of people for scenario 2, their gut reaction is it's not a cure because it's a thought experiment because you're not actually kind of producing any real data. But I would agree that it depends on how we're defining thought experiments here. If you're a theoretical physicist, your thought experiment is the experiment, right? If your, discipline is building theory, right, as what's truly novel, as what's pushing the boundaries of the discipline, then I would argue that that's a cure. Now that's harder to do with a novice student, right? And so that's something to think about, right? Especially if you want to build these into intro courses, how do you do that with students who come in knowing very little?
I just want to, so we did our department was revising our curriculum for biology, and we went through the vision and change core content competencies, and one of the things that our faculty, many faculty did not think was important was the modeling, being able to do predictive modeling, and it's because most of our faculty come from a strict experimental background, and so they would assume that those folks would say thought experiments aren't real experiments, and it's because of this disconnect between the actual experimental design and the thought experiment that modeling does. So that language is a disconnect even among faculty, let alone our students.
Yeah. So then so for that one, I would say, it depends on your discipline or even your subdiscipline, right? So, so I teach an animal physiology course. In my lecture course, I have students do what I would consider thought experiments all the time, right? Brainstorm, what you think a new treatment to diabetes might be. That is not at all a cure, right? There's no way that that's going to lead to something broadly relevant. But if in that discipline, that question that you're having students grapple with could actually push that boundary of knowledge, then I would say that would count. What about 3?
It's the same thing, like, it's like the doorknob experiment, but it depends on the design we thought, because you might have a novel way of looking at how the experiment is going to result in finding something new, whether it will help advance knowledge or not.
Yeah, so I would say most of the time that type of experiment would be considered more inquiry, not a cure. Right? It could be, again, super interesting for the students, super motivating for them. They could figure that out. I've had people argue that it could be a cure if the students are walking in some weird place, then they could actually identify a completely novel bacteria from their shoe. Then that could be a way to actually turn it into a cure, but that's probably going to be less likely, right? So I would say most of the time scenario 3 is probably not going to be a cure, unless students are, walking in Antarctica and identifying some bacteria that has never been identified before. But it's probably not going to happen here that much. And then what about 5?
We thought 5 was a cure, and we weren't sure that you would publish a paper that was like, here's the set of mutants that we made, and they all have the same activity as a native protein.
Yep.
But we could imagine that data being really useful in a report or a database that could then be built upon.
Yeah. So this is a point that I really want to make, that it matters if someone outside the course cares about the results, and failed experiments and negative results are a part of research, right? And so it's that potential, it's the design of the cure needs to be designed in a way so that someone could build upon the findings, that the findings could impact people's behavior, that it potentially could result in some kind of community report or publication. But the reality is that most cures, particularly the first time, the second time, the third time it gets taught, it's not going to happen, right? Either the science itself is not going to work, and you're not going to publish a paper with negative results, or there's going to be a ton of logistical things that you're going to have to work out, right? And it doesn't mean that that's not a cure, and that what the students are experiencing is not a cure. It's just you have to work through the tweaks of that, right? And so, when I think about kind of conceptualizing what makes a cure, I think about it from the design perspective. So the instructor, the faculty member, or the teaching assistant, or whoever is in charge of actually setting up the cure in that design, are they designing it with the intention that someone is going to care about it outside of the course?
So I find negative results are often uninspiring to students. So I guess I'm curious how you would design a cure that you anticipate may have negative results, that you can still, otherwise are you like now losing students that would have gone into research because they're like, this is so awful.
Yeah. I think you're hitting on something that's super important. I think that most people would say failure is a part of cures, it's awesome, students can learn so much from failure, it's authentic failure, it's not like getting a C in a course, like this will prepare them for a more authentic view of what research is, but I agree completely with you that that could be really disheartening for students. And when I've taught a basic science cure, that has led to kind of negative results, super disheartening for students, right? They're like super disappointed at the end. They want to be able to see something. I've actually had a situation where some of the students' experiments, they basically were working on mutant versions of the same protein. Some students showed an effect, a phenotype, some did not, right? And so even within the same class, we saw kind of differential impact on the students. And the ones that didn't have a phenotype, for me, I was actually super interested in that. I thought, you know, why? Let's figure that out. Like, and it was much harder to get the students motivated. So that said, I think that there's kind of two camps right now. Some people are like, it's great, students are going to experience failure, it's going to normalize failure, etc. Cures can be this great opportunity for failure, but I think you're spot on that failure can be really disheartening for these students, and maybe they won't want to go on and research, right? And so I think that becomes a little bit of an ethical question. Do we want to pretend to students that science works more than it typically does, right? Do we want them to get more excited about it? And maybe we do, right? And I know, and I've done this in the past, and I know some colleagues that have done this in the past, where maybe what you do is you do your preliminary experiments yourself, right? Or with graduate students in your lab, and you see interesting kind of first steps of things, and then you bring that into the lab with the students, and so you know that that first experiment will show some kind of effect, right? And then they can do the subsequent steps, right? So it's a little bit less like completely real research because you've done that first step, and then they're almost replicating that first step, but that can be one way. So you can almost screen your projects first to see if something's going to fail or not. Or you